Welcome to our webinar for the Globe North American Fall Phenology Campaign. My name is Jennifer Bourgeot. I'm the United States Country Coordinator for the Globe program. We're going to tell you a little bit about Globe once we get started, for those of you that are unfamiliar. And right now, we are really excited about all the mission connections, the NASA mission connections that we're making to the Globe program, and this is one of them. So for years, we've had a phenology campaign, and we have been collecting data for a number of years. Particularly, I want to shout out, because I know they're here, our colleagues from Alaska, and their people that they work with. And then our colleagues from Maine have been superstars at the Phenology campaign, as well as a number of teachers across the country. So welcome to everyone. My colleagues here are Alicia Carlson and Haley Wickline. We are all in the Northeast. We work through the University of New Hampshire. And so what is the Phenology campaign? If you are here, you probably know a little bit about it. The fall campaign is from August to December, and what we're tracking is the changing of the leaves. In the spring, we do this from February to June. We do this for a long period of time because we're from all different parts of the country. And when you are participating in the Phenology campaign, you are joining a large network of citizen scientists, all the way from pre-kindergarten up to adults that are tracking the spring and the fall. If you don't know what GLOBE is, it is a program that is sponsored by NASA, and it provides the public worldwide with opportunities to participate in Earth science data collection and the scientific process. So if you look, we've been in existence for 30 years, over 30 years now. We have 290 million measurements. The Green Up and Green Down campaign is one of about 40 measurements that we can take, and that citizens around the world can take. So we have many people participating in over 127 countries. Green Down is one that we're particularly excited about. It is, I will admit, my absolute favorite season of the year. The colors in the Northeast are phenomenal. And so it's also my birthday kind of time of year. So, you know, it's maybe, I don't know, might just be my birthday. So Green Down marks the end of the growing season for many plants. There is a leaf color change because of the breakdown of chlorophyll. And the colors, if you know anything about New England, we have lots of different tree species, and the color progression is very different for those tree species. This is also called senescence, and it is initiated when we have fewer hours of sunlight, lower temperatures, or in the other areas of the country when it's drier or warmer temperatures. Okay, so this year there's two ways to participate. And Haley, is this you? Nope, I'm going to keep going. There's two ways to participate this year. The way we've been participating in the last couple years is monitoring the leaves on individual trees. And that is using the Green Down protocol. You're tracking 4 leaves on an individual tree. So this is great if you want to focus in on one tree on your site. For instance, if you're at a library and you have a tree there, then your patrons could be monitoring one tree. Or if you're lucky and you have a couple different species, it's a great opportunity to compare how those trees are changing and how it's different between the different species. And then really exciting this year, we have another opportunity to contribute in a different way, and that is to take landscape photos for the NASA's PACE satellite mission, and that's using the land cover tool in the Globe Observer app. And we're going to talk you through both of these ways to participate. And we're also going to hear from the PACE scientist who's needing this data. And he's going to share what he's going to be doing with it and what he is studying. The PACE satellite launched in 2024, and it is giving us a new view of leaf color change. We've been able to see it with other satellites as well, but this is a new exciting way to look at leaf color change. And the NASA scientists are working and they're asking for your help collecting ground-based observations. So with that, I'm going to turn it over to our PACE scientist, Fred Huemmrich, and he's going to talk to us a little bit about the science behind that and what he's seeing and studying. So I'm going to stop my share.
Well, thank you, Jen. Um, so let me just say something about PACE to start out with. PACE stands for Plankton, Aerosol Clouds, and Ocean Ecosystems. And as the name implies, it's really a mission that is focused mainly on studying the oceans and the atmosphere. But I've been using it to study land surfaces, and it provides a really unique um, data set because it gives us hyperspectral data, which means lots and lots of spectral bands. But it does this globally every 2 days. It covers the entire globe. So we get repeated observations like this, and there is no other satellite that provides. There are other instruments like emit on the space station, for example, that can do this hyperspectral measurements, but they only do it in small areas and they have to be tasked to do a specific place. PACE does the entire planet over and over again. And this gives us an opportunity to look at the world in a different way. And in particular, I've been looking at the leaf color change in autumn. And so, as Jen said in the introduction, uh, uh, and you probably already know, but let me just go over this again. What happens in the fall is on deciduous trees, the the tree breaks down the chlorophyll that's in the leaf to and reabsorbs those nutrients so it doesn't lose them when it drops the leaf. And so that takes the green color out of the leaf. And then when that green color goes away, other chemicals in the leaves, other pigments called carotenoids, which are already in the leaf, become visible, and those tend to be the yellow kind of colors, and so they're already in the leaf. And then the third color in that comes in here is anthocyanins, which are red pigments, and anthocyanins are actually actively produced by the tree in the fall. um, no one knows exactly why the trees do this. It may help to for the reabsorption of the minerals or the nutrients, and it may have something to do with protecting the leaves from insect damage during this period. But anyway, it happens. So the thing that is interesting about PACE and where PACE comes into this is because PACE has all of these narrow spectral bands, we can calculate vegetation indices that are related to the amount of these pigments in the plant canopy. And this is the first time we've been able to do this again over large areas and repeatedly. And so there's a chlorophyll index, there's an anthocyanin index, and there's a carotenoid index. And these are using specific narrow spectral bands that have been shown to be sensitive to those pigments in the leaves. So, we're talking about this is the whole name of this program is Green Down, and most of the remote sensing studies of plant phenology have looked at greenness indices like NDVI or EVI. And the plot here is showing kind of a typical NDVI seasonal curve for a deciduous forest. It greens up in the spring, it's sort of flat through the summer, and then it drops off in the fall, where the area I've highlighted. And the thing that's the problem with this is that that greenness index is affected by both the color of the leaves. If the green leaves change color, that affects it, and the number of leaves. So, because the trees are both, the leaves are both changing color and it's dropping leaves, and we can't tell the difference in that in this curve. And so, what generally happens if you want to define something about fall colors or the change in fall, you just arbitrarily pick a point on this curve, like halfway down or something like that, and set that to your value. But it's an arbitrary thing. But now with PACE, we can look at these pigment indices. And so now we're seeing the pigment indices for the anthocyanin as an example and the chlorophyll. So the chlorophyll is the green curve. The anthocyanin is this kind of purpley curve. And the ratio of the anthocyanin index to the chlorophyll index is that red curve. And that's the one I really want to focus on here because what you see is during that fall period, we see this peak occur. So we actually can have like a real point in time that we can point to as being the time of peak greenness. And as we want to look at phenology change over time, as PACE collects more data year to year, this gives us a real marker that we can track. And the reason we do that ratio between these indices is basically what these indices are related to is the amount of leaf pigment, in the leaf and the leaf area. And so by taking the ratio of these two indices, we kind of cancel out the leaf area, at least minimize that effect, and highlight the change in the pigment pools, which is affecting the leaf colors. And so that's what we're doing here. And so we can so then the question is, so we can map these pigments, but is it what we see? Does it affect what we're actually observing? And so to check this, what we've done is we've looked at the phenocam data, and I don't know if you're familiar with phenocams, but there is a network of web-based cameras mounted on towers in places all over, well, literally all over the world. And it basically just take pictures, color pictures regularly, like the pictures you see here. And what's usually done if you look up the phenocam, and if you haven't, I urge you to do it because it's enormous fun. They calculate what's called the green chromatic coordinate, which is basically the green gun from from the camera divided by the sum of the red, green, and blue colors intensities. And and that basically gives you it's kind of like NDVI in a way. It gives you a mark of like the green color. But what we want to do here is we want to look at the color leaf colors. So we've changed it to being the red chromatic coordinate, the red value divided by the sum of the three. And in the plot here, that red line is that red chromatic coordinate from a phenocam from this site. And the blue line is what we're getting from the PACE data. And you can see that the red peaks up. So our perception of red, is, at least according to the camera, peaks up here, and it matches the timing of what we see from PACE. And we've done this for 6 different sites scattered around the eastern deciduous forests. And both of these plots, the solid lines, which are identical on both of the plots, are the is that red chromatic coordinate from the phenocam. And there is like there's like almost a month difference in the timing of peak redness in these sites, but I've aligned them all so that the date of peak redness is at 0, and then we compare that to our anthocyanin index from PACE divided by the chlorophyll index and the carotenoid index divided by the chlorophyll index. And you see that they all are peaking up along with the phenocam. So this tells us that we're seeing something. And this gets to what I really want to observe more of is to see these kind of images taken in a lot of different places, just a handful of places, to try to make sure that we understand what the satellite's actually telling us. So we can take that data. So now we have the satellite data, and so this is data from the PACE satellite, where at the start of the 2024 fall season, the end of September, beginning of October. So this is an 8-day average, and it's this anthocyanin index, the red pigment divided by the chlorophyll index, is what's being mapped here. And so basically, things that aren't blue are showing us fall colors. And here at the start of fall here, we're seeing, leaf color change in, kind of northern mountainous regions, the Laurentians, the Adirondacks, the Poconos, the White Mountains. And then we can step through the time. So again, because PACE makes these repeated observations, these are 8-day averages, and we can step through and watch the progression of fall colors through it. And now at this point, now that we're at the end of October, you can see that down the Appalachians are in peak fall color, and those northern sites have passed peak. And so the values have dropped off, and we can watch the fall colors travel down through there. So one of the things that I found it was really interesting when I first did this is that if you look at the onset of fall colors, they actually define ecoregions. So on the left side here are maps of ecoregions based on vegetation type, tree types. And you can see how the shapes of those match up with the onset of fall colors we see for the Adirondacks, the Catskills. We can also see it, excuse me, for the unglaciated high Allegheny Plateau. I have to say, I grew up in Western Pennsylvania, and I didn't know this ecoregion existed until I mapped it here. So this is an interesting thing in terms of exposing uh, specific ecoregions out of this data. And then we can see again back at the Adirondacks that now we're moving further down. So now we're into mid-October, that the peak of the Adirondacks is past peak redness, but the foothills now are turning color. And then this is one of my favorites up here in the north at the Hudson Bay. This is not a deciduous forest at all. This is a wetland in the boreal forest, and it turns on this really bright, shows this really high anthocyanin content there. And one of the things that's interesting about this wetland in particular is it's one of the major methane sources in the boreal forest. And so this approach, although it wasn't intended for this, is actually highlighting this interesting ecoregion. So we can use this data because PACE continues to collect data, and this is for the 2024 and 2025, showing the end of September and into the mid-October. And you can see the upper row are the data from 2024, and the lower row is data from 2025. And you can see the similarities and differences as you go from year to year, particularly like, for example, look at the end of October, the far right ones. You can see the big difference in how much the fall colors have expanded in 2024 compared to 2025, looking down the Appalachians. And then the other thing, again, I've been just showing the anthocyanin index divided by the chlorophyll index, but there's also the carotenoid index, the yellow pigments, divided by the chlorophyll index. And these are for the same dates. And you can see that there are differences in like where we see the fall colors, but depending upon which index we're using. And that's going to depend on species. Some species have more red in them than others, and also due to climate conditions, because depending upon the environmental conditions, since the anthocyanins, the red pigments, are being generated by the plants, they depending upon the weather is going to affect how much you get of that, and then how much we would see. And then here is another example for a little bit later in the season, comparing the two. Uh, and so basically, just to end this, there's lots of good reasons to be able to map and study these the timing of and the distribution of autumn colors, going from studying ecosystem productivity and energy balance, but also there are economic reasons because tourism for fall foliage is an important thing. Pace's observations give us a new view of this that's really more physiologically based than those previous greenness index based studies, and gives us this description of leaf color change and so in terms of these pigments. And so if we can tie this to actual views of leaf color change, then that's going to firm up our understanding of what it is we're actually seeing from the pace data. And so just I want to thank NASA for their support of this, and for the NEON, the National Ecological Observatory Network, which supported the phenocams, and the phenocam network, which provided the data that I used for the validation. And thank you.
Thank you. That was awesome. And now I'm really excited to provide this data. Now I'm going to go back to sharing my screen, and Haley is going to take over, and she's going to walk us through the steps that you would be doing in these two different ways to collect the data. So, Haley?
Great. Hi, everyone. My name's Haley Wickline. Thanks again, Fred. This is really exciting. We're so happy to be partnering with you this year. As Jen said, I'm just going to walk through the specific steps for the two different ways that you can help us collect phenology data this year, and you can choose one or both to participate in. So I'll start out talking about these landscape photos. Whoop. There we go. Start out talking about these landscape photos that the PACE scientists are looking for. To use these, you will download our app, the Globe Observer app, if you don't have it already, and create a free Globe account. And we'll stick the link in the chat of where you can get that app. So you can download that onto a phone or a tablet. And then you're going to choose a location where you can see the forest landscape. So PACE is giving us this really broad view. We're zooming in a little bit further, but not all the way to like one tree. We want to be able to see some nice forest landscape in our photos. These can be a one-time thing. Maybe you're hiking or you're leaf peeping or taking a trip somewhere, and you see a nice forest view, and you want to take some photos, or these can be repeat measurements. So maybe near where you live or where you work, there's a good forest view, and those repeat measurements can help us see how that landscape's changing across the season. But either one is okay. Once you have the Globe Observer app and you log in, we're going to be using the land cover tool. So you'll open that up. The land cover tool has some in-app training, so it'll walk you through how to take measurements with the tool. And then you'll answer a couple of questions about the ground and sky conditions. So just kind of yes or no, is it raining? Is it snowing? Is the ground dry or wet? That kind of thing. After that, it will prompt you to take photos in each cardinal direction, so north, south, east, west, as well as one facing up to the sky and one down to the ground. And the app has this really neat, these little circles. So you see in the picture under number 2, that circle with an N in it. So I was facing north when I took this picture. So the circle just kind of bobs up on your screen when you're facing the right direction, and you line it up with that gray band. And then you just tap anywhere on the screen and it will take your photo. So you'll do your 6 photos, north, south, east, west, up and down. And then it gives you the option to take a feature photo. And this is the really important part for participating in the Phenology campaign. You're going to face wherever you see the most color in the forest landscape. So now you can face any direction you want. And again, you'll tap anywhere and you'll take that feature photo. The one thing that's really important when taking these photos is that we don't have faces, people's faces, identifiable people in them, because we aren't able to use those. So just make sure anyone that's with you is out of frame when you're taking the pictures. The one thing I will say is sometimes it's really hard to not get your feet. When you're doing that down photo, you have to like stretch your arms out really far. So feet are fine. If you end up with your feet in your down picture, that's okay. After that, you have the option if you want to analyze your land cover, which is estimating the percentages of different types of land cover you're seeing in your site, wherever you're taking your photos. Or if you're just trying to really quick get those photos for the PACE scientists, you can click the gray button, which says skip analysis and save my photos. And then the very important part is on the next screen that you click send all land cover data now, because that's what will submit your photos to Globe so that we can access them. The nice thing about the app is if you're in a place without signal, cellular signal or Wi-Fi, it will save your photos in the app. And then when you get back to a place with signal or Wi-Fi, you can go back in and submit your photos later. And I will just say one tip, which I just learned, which is that I was out the other day doing my landscape photos and I was on like the teeniest amount of Wi-Fi and the app started freezing up on me and I couldn't figure out why. Um, and the trick is, if that's happening, is to turn off your Wi-Fi because it was trying really hard to do everything over like barely any Wi-Fi. So if you put your plane in, or not your plane, your phone in airplane mode, or you're turning off your Wi-Fi, take your photos, and then when you're back near your building or your house, you can send the photos. And that's it. So it's actually, once you get going, it's pretty quick, just taking a couple of photos and hitting submit. So that's number one. And then we have another way to participate. So now we're going to zoom in even farther and get just to that individual tree, individual leaf level and take a look at how leaves are changing color across the season. So for this way of participating, you will also need a Globe account to submit your data. You can make your account in the Globe Observer app or online on our website. And it's the same account. So once you have one account, you can log in online, you can log into the app. You're going to pick a site that is something you can visit regularly and choose one tree to monitor. So for this, it's great if you can to choose a site that's a little bit away from buildings and not super watered and fertilized. If that's not possible, also totally fine. Still feel like you can participate and be a part of this. There's a place when you're entering your data, you can just make a comment about where your tree is. What is really important though is that it's an accessible spot, safe to get to, and that you can actually reach the leaves. So you don't want to pick one of those really tall, beautiful, canopy up high sorts of trees. You want to be able to physically reach the leaves to collect your data. You'll choose one branch and you're going to mark that branch with flagging tape or a piece of ribbon so you know where to go each time. And then you're going to identify your tree species. If you aren't an expert at this, that's okay. There are lots of different resources out there to help. Lots of apps like Seek or LeafSnap, iNaturalist, or I actually bring my old-fashioned paper ID guide out with me, so you can do that as well. Once you have your branch, you're actually going to tag 4 leaves to monitor through the fall. So you're going to find the leaf that's at the end of the branch. That's called the terminal leaf. That will be your leaf 1. And you're going to label it by marking 1 dot on the branch. So not really on the leaf, but right on the branch next to that leaf stem. I just use a black permanent marker to do this. We've had people use whiteout. I had a teacher tell me silver permanent marker shows up if your bark is dark, but whatever works for you is great. So you'll label that one with 1 dot, and then you're going to find the 3 leaves closest to that leaf and label them with dots as well. So leaf 2 has 2 dots, leaf 3 has 3 dots, and so on. So then you have your tree, your leaves are tagged, and you're ready to go. To measure green down, visit your tree. We recommend at least twice a week. More is awesome. If you can only get out there once a week, still get out there and do it. That's okay. At each visit, you'll use the Globe Plant Color Guide to estimate the color of your leaf. So in this picture here, this is our Phenology ruler. So we have the color guide on one side, and then the back side is actually a ruler. So if you join us in the spring, we're measuring the leaf with a ruler as they leaf out, and you can use the tool for both spring and fall. We also have printable versions on our website, or if you're going out with the Globe Observer app, the color guide is in the app. So lots of ways to get that. We're looking for the dominant color. So we all know as leaves change color, sometimes you see lots of different colors on one leaf. So just pick the color that's kind of the greatest percentage. And underneath, you can kind of just barely see in this picture, there's text under all those little color boxes, and that's a Munsell color code. So if you're recording your data on a paper data sheet, you're just going to record that color code for leaves 1, 2, 3, and 4. If you're in the app, there's just a little checkbox you'll hit once you've chosen your color. And then again, super important that you upload your data. So you can do this on the app. We also have online data entry, so you can do it through your computer. And we have tutorials available for that. We actually have a tutorial on our website that walks you through just entering green down data, so you can see exactly how to do it. And then Globe has some great visualization tools. So once you enter your data, you can actually see it on a map. You can print out a graph of that color change. So it's really awesome once your data are in there. I will just make one note that if you're working with groups of people, so maybe you're a librarian and your patrons are helping you monitor your tree, or maybe you have, a classroom of youth that are going out and monitoring a tree, and they're collecting data on the same leaves. We just recommend that you're the one that enters it. So we keep all that data for that tree kind of in one place and we can see the change. So those are the steps, 2 different ways to participate. We have lots of resources and tutorials to support you through all of this. And I'm going to pass it over to my colleague Alicia, who is going to walk us through the website and where to find all those things.
So I can walk you briefly through our website. I know Haley's going to put the link to the website in the chat there. This right here is our campaign homepage, and this has the details that you need to know to get started.
Coming over at 4.
And so you can see just at the top here, there's some really basic information. There's this new to Globe. So if you need to create an account, there's a link there to help you to do that. It's a pretty easy process. There's tips, real quick tips on how to monitor the leaves on an individual tree. Scroll down, take landscape photos. And then we have some resources here for different audiences. So if you're an educator in a classroom, or you have an after school group or something like that, we have these 2 green down sequences. One's for K to 8 levels and the other one's for 9 to 12. And it's just like a step-by-step process to help you do green down with those different audiences. And then for our informal participants like librarians or science centers, we have a toolkit, again, that's the step-by-step doing each part of the process and working with your different audiences. Then we have some PACE resources that I'll show you in just a moment. And we have this campaign media kit that's a Google folder. It's got a flyer and some sample text for posting on social media and some imagery. So if you are doing this at your facility and you want to get people interested, you can use any of those materials to get started. So back up at the top here, I will say right now you can see this sidebar on my screen. If you're looking at this page on a phone or a tablet, or if your browser window is just slightly smaller, you might not see the sidebar. It might appear as a drop-down menu. So just be aware of that. So these protocol tutorials, Haley was just talking about, we have these really short videos. They're like 3 minutes long at the most. Again, on the different steps to go through these protocols. So the first one is selecting a site and monitoring your tree. So if you didn't understand what Haley was saying about how to put the dots on the tree, she will show you how to do that in this video. And then we have the using the color guide, which I think is pretty easy. And then the uploading your data to the database. So again, very short videos, really well done, if I might say so myself. Okay, and then I'm going to skip over the plant color guides, but there's links there you can download if you don't want to wait to get a ruler, then you can go there and print some off yourself. But here's this PACE resources webpage. It has some really basic information about the PACE mission at the top. And then there are some fun visualizations and videos that you can use to get your different audiences interested in the program. And at the bottom here, some related Globe learning activities that connect well with the PACE mission. So there's another page of learning activities. They're all, again, Globe learning activities. I'm going to skip that one though. If you're working with audiences that are interested in career connections, so if you're trying to get youth interested in science careers, we have on this page 8 videos from different scientists that study different aspects of phenology. They're short. They're about 5 to 10 minutes long. They all answer the same 6 questions, but in very different ways. So we recommend these for those of you that need a career connection. Somebody asked in the chat if we're going to be sharing these slides, and we will, and they'll end up on this webpage, the Phenology webinars. You can see some past webinars here. That one's for the spring. There's this great webinar we did a couple years ago with 2 educators where they shared their ideas for doing green down in their different classrooms. And then we have this page just of so many different resources. Haley talked about needing to identify your tree. There's a section on that that includes some links to some good resources for that. There's one on the science of fall colors, which is interesting. And then again, here are these green down sequences and the informal educators toolkit. And then I really would recommend, I'm not going to show these to you now, but there's these wall displays that one educator did with her classrooms. She had multiple classrooms looking at multiple trees, and they took like a big sheet of chart paper and posted it on the wall and monitored their color change throughout the season on all the different trees. So it's a really nice visual way for everybody to see what's happening. And that's all I'm going to share from here. So look at the website. I skipped over so much stuff. You'll find everything you need there to help you get started. And Jen, can you bring back up the slideshow, please? Great. Can I move it? Oh, there we go. Okay, so for those of you in the United States, whoops, we can send a kit to you. And that would include the phenology rulers, a data sheet, a field guide. So that's like the quick steps to doing the protocol. If you're an informal educator, we'll send that information packet, the toolkit. And for formal educators, we'll share those sequences again with you. And so Haley's put the request form in the chat for that. And finally, it's taking its time to move, but I know it's going to do it eventually. So here are just a couple QR codes, again, for our campaign webpage. And if you want to get emails, we're going to send fairly regular emails throughout the season to our phenology list. And so you can sign up for that either with that QR code, or I see Haley just put the link in the chat for that too. And that's all I've got. So I think I'm going back to Jen for the last slide here.
Yes, you are. Sorry, I was typing and answering questions in the chat. I just want to, we're getting questions again about from different groups about how you can manage this, and especially with if you have library patrons or if you have schools and there's privacy issues, etc. So just to clarify that one question. So with the landscape photos, you do need to do that with the app, the Land Cover Observer app, the Observer app, sorry, and the Land Cover Protocol. So those would be the landscape photos. If you wanted to just do the leaves, we do have paper data sheets available for that. So you can take those paper data sheets out into the field, enter all that data, and then you can enter the data that you've collected on the paper data sheets through your computer. So there is a data entry portal through the Globe website for you to enter that data. You can also have students create their own account if they're over 13 years of age. So if they have access, or you can send off information about the Globe Observer app to collect the data with their families. So their families can create a citizen science account and they can also enter the data through that citizen science account. We have something called guest accounts. So if the issue is a privacy issue about your students getting an account themselves, you could make an account and then you can create a guest account and have students enter the data using the Observer app through a guest account that is under you as an educator or as a librarian or just as whatever facilitator you are. And so that's a way to address the privacy issues that you may be facing. I just want to take another opportunity before I say thank you and goodbye. That we have another opportunity with PACE and Globe, and we have a Globe Clouds Challenge coming up between September 15th and October 15th. And there's going to be an upcoming GeoConnect webinar about that challenge coming up, and then there'll be a training webinar on September 9th. You can always watch this over again. We did go quite quickly. There's a lot of good information in here. So hopefully as this challenge goes on, as our Phenology campaign goes on, we will be giving you opportunities to interact with us. If you have any questions, we are happy to answer them. We are going to put an email address in the chat, and it'll also be in this webinar and in our follow-up for you to ask any other questions that you have. If you need one-on-one support entering data or figuring out the protocols, we are here to definitely support you with all of that. And we will get any answers that that we can't answer ourselves. So thank you, everyone.